Hi guys, I'm Nidhi, a student from Cardiovascular Technologist. So today I and my group mates are here to present a topic about aberrant brain connectivity in congenital heart disease before cardiac surgery. CHD is an abnormal condition of the heart present at birth. It can relate it to ineffective pumping of the blood to different part of the body, which can lead to ischemia and cyanosis. So the research was done by a group of researchers. It was done by Children's Health System Network in Washington DC. The study was conducted on 16th of June in 2016. In this study, the researchers had taken MRI images from previous interaction with the newborns. They have not tried to influence the data or result by any means. This study was conducted because no other study was conducted previously regarding functional brain connectivity, which is altered in high risk population after birth. The objective was to compare brain functional connectivity of resting networks in healthy newborn with full term newborn with congenital heart disease. It was done using advanced neuroimaging technique such as MRI data. These MRI scans were done on control group and the CHD group. Parental consent was taken and the study was approved by Institutional Review Board. Mentioning about the variables, then the study variables were mostly quantitative, such as age at birth, weight, APGR score, oxygen saturation, pH values, etc. APGR score is the score given to the newborns depending upon how healthy they are. Some qualitative variables like gender, multiple cardiac dysfunction were also noted. The type of CHD is qualitative variable though. At the beginning of the study, 138 term children were chosen, 43 who had CHD and 95 healthy record control children. Unfortunately, 23 subjects were removed from the set. The rest 115 subjects were studied by an expert neuroradiologist. Two of these subjects had brain injury. One had underdeveloped cerebrum, who were then also removed. That brought their actual sample size to 112 neonates. Now, their main objective was to compare whole brain functional connectivity of resting state network in healthy term newborns whose number n is equal to 82 and full term neonates with CHD before surgery whose sample size n is equal to 30. They were done by using graph theory and network-based statistics. So now the further details would be provided to you by Abu.
Thank you, Nidhi. So, as my friend Nidhi indicated, researchers were using data from samples to come into a conclusion. This is the definition of an observational study where researchers gather the data from samples but do not influence the study in any way. Here, our researchers they were gathering data from MRIs and other imaging data of the brain and the heart to come into a conclusion, but they were not taking any steps to create a positive or negative outcomes. Throughout the study, the researchers used terms like rich club node and small world network. Even though these terms were used over and over again, they were not defined in plain English for the common people like us. So we had to take it on our small, little, tiny shoulders and get out there to get some answers. Hashtag Google search. We found out that Rich Club is not some club for hairless billionaires like Elon Musk and Jeff Bezos. These are regions of our brain that plays a major role during information transfer. They require more energy to process data, but it is also very efficient. These reach club nodes showed in higher degree in control group than neonates with CHD. On the other hand, a small world network is where most nodes are not neighbors to one another, but most nodes can be reached from every other node by a small number of hops or steps. So they're still connected, they're just not next to each other. Now, one thing we must remember that this study is one of its kind, and it discovered something surprising. It found that while there was evidence that brain connectivity of CHD neonates was poorer than normal control newborns, the changes in connectivity of CHD neonates are still not very clear. And the local network, their small world networks, were still functional and quite efficient, which creates an opportunity for early medical therapy and future intervention if needed. Now, I will hand over the mantle to my good friend Joseph, who's going to talk about.
Hello everyone, my name is Joseph Jespin and I'm here to pick up from where my friend Abu left off. So, we have learned so far that the study was conducted to find the relation of CHD to the functioning and development of brain in newborns. So, what did the researchers find out? To the surprise of the researchers, they found out that even though there was reduced blood flow to the different parts of the body and brain in the CHD group, the ability of the brain to maintain its function and transfer impulses at a low cost energy was still maintained. But there was other limitations to the CHD group when compared to the control group. As my friend mentioned before, we can see that the rich club nodes in the CHD category is limited when compared to the control group. It's also noticeable that the connection between these rich club nodes is also limited in the CHD group. So why does this matter? The transfer of impulses from brain happens through multiple channels, and these rich club nodes are the major connections. They have like a ton of connections. They are like the international airports which connect the local airports and the international airports. So the transfer of impulses are kind of affected. So what does this suggest? This suggests that the development of brain in the CHD group is affected due to the reduced blood flow and all the things that are happening during a CHD. We can also see that the researchers have found out that the subcortical region of the brain is highly affected in the CHD group. The subcortical region of the brain is what is responsible for the cognitive functions like attention and everything. So this may lead to deficit of attention in these children. What were the limitations of the study? The major limitation was that the sample size and the CHD group that was involved couldn't properly represent the population. It's mainly because that the sample size was too little, and also the CHD is a classification of disorders which is present at birth. It can range from ventricular septal defect, which is like a opening between the two ventricles. There is an opening in the septum, to a very serious condition, the transposition of great arteries, in which the aorta and the pulmonary arteries are switched. So we can see that the seriousness of these conditions are different, and the effect and cyanosis that these conditions causes can range between each disorders. So we can't be specific that the this sample that we took is not depicting the entire population. A much vast study with a much bigger population and a similar congenital defect is required for a much better response and result. Another drawback to the study was that the researchers were recording the resting state of the brain in these neonates. And 3 of the neonates in the CHD category was under anesthesia when this procedure was done. Why does this matter? It's because that still today there was no study that was conducted to find the effect of anesthesia on the brains of the newborn neonates. So, why does the findings matter? Since the study was done using fMRI, the difference in the brain functions that were noted in the control group and CHD group was representative of the effect of fMRI in determining the early diagnosis of the brain conditions in the CHD category. So it was found that this test can be used in the early diagnosis and treatment of this high risk category. So have a nice day. Thank you for your precious time.
Testing 1, 2, 3. Testing 1, 2, 3. Testing 1, 2, 3. Testing 1, 2, 3.